Let me go to our next speaker, Professor Donald Royall from University of Texas Health Science Center, who will talk about probing the biomarkers of dementia disparities with structural models. So go ahead, Dr. Royall.
Hi, good morning. I want to thank the organizers for inviting me to give this talk. I'm chief of the Division of Geriatric Psychiatry at the University of Texas and have been working on blood-based biomarkers of dementia severity. So I'll be talking about using structural models to probe disparities in demographic differences in dementia and trying to identify their biomarkers. I don't have any meaningful disclosures, but you might be interested that I've incorporated a company to leverage some of these insights, but right now it has no investors and no sales, so it's something of a paper tiger. Anyway, there's obviously a great interest in demographic disparities and Alzheimer's risk. In parallel to that, my group has been developing structural equation models to identify mostly blood-based protein biomarkers of dementia severity. And what I'd like to do is identify the biomarkers that specifically mediate individual demographic risks, propose interventions on those biomarkers, and then select individuals who would most benefit from treatment on the basis of having a risk to have been adversely impacted by the biomarkers of interest. I'll be showing you data from two data sets. The first is the Texas Alzheimer's Research and Care Consortium. I'm the PI of the San Antonio site. TARC is a large, descriptive, convenient sample, about 3,500 well-characterized volunteers. It's ethnically diverse. 35% of the cohort is Mexican-American. They've been followed prospectively. This is a little bit old now. It's probably closer to 6 years now. We have a comprehensive psychometric battery available in both English and Spanish at the convenience of the subject, and a panel of about 120 serum protein biomarkers provided by the Myriad human map profile. It's a multiplexed immunoassay profile. And in TARC, these proteins are being measured in serum. We also have access to the Alzheimer's Disease Neuroimaging Initiative, or ADNI, which is a similar large descriptive convenient sample, but lacks ethnic diversity. TARC was actually modeled on ADNI, and so there's a great deal of overlap in the psychometrics obtained in each of these two studies. And we have the identical protein biomarker panel measured by the same vendor, but in ADNI, the proteins are being measured in plasma, not serum. And the foundation for this work is a dementia-specific phenotype I've developed, which we call Delta, or D for short. And I'll give you a little bit of background on this. So D is a latent variable constructed in a structural equation model framework. It represents or captures the cognitive correlates of functional status. And the idea of this is that dementia is not really cognitive impairment, it's disability arising from cognitive impairment. Non-disabling cognitive change is really orthogonal to dementia severity and doesn't contribute to dementia. So basically what D does is provide a completely empirical assessment, completely quantifiable assessment of dementia's essential cognitive feature. In theory, D can be derived from any battery, any cognitive battery that contains a measure of instrumental activities of daily living. And we've explored that in various papers all the way down to the item sets of a single test. In practice, D has been derived from dozens of cognitive measures. So it really doesn't depend on which cognitive measure you have available. 17 D homologs have been validated to date. And I'm borrowing a term from genetics here. A homolog is a gene descended from an ancestral gene in the same species and that retains the original's function. So when we operationalize a D homolog in a particular battery, it may, the measures involved may not overlap at all with some other D homolog in a different cohort or data set. And yet D's psychometric properties are very stable and robust. So, so far, D has been validated by independent groups. It's not just my group. In 4 countries on 4 continents, in over 30,000 subjects with different ethnic, educational, linguistic, and cultural backgrounds. And across all these studies, D homologs are strongly related to dementia severity as measured by clinicians by, for example, the Clinical Dementia Rating Scale Sum of Boxes, which is available in many different studies. So we use that kind of as a touchstone to validate different D homologs against. And D achieves an average association with the CDR of 0.83 across 18 published models. D's ROC for the distinct discrimination between Alzheimer's cases and controls in these various studies ranges from 0.92 to 1, with a mean of 0.97 across 19 published models. So regardless of the measures being used to construct the construct, it is strongly related to clinical severity as rated by clinicians and achieves a very high accuracy of discriminating dementia from non-demented states. So what D does for you is pretty interesting. It converts dementia from a category to a continuously distributed omnibus dementia severity measure. And it also happens to be agnostic to etiology. So I'm proposing that D is dementia's essential cognitive impairment, that no disease process or biological process is associated with dementia unless it can significantly impact D scores. And this is true across etiologies because fundamentally they're all dementias. They all must share dementia's essential clinical feature. It's constructed by a unique bifactor model and a confirmatory factor analysis that's relatively easy to do and straightforward. And the latent variable can be output as a factor score, a D score, that can then be applied to individuals as a dementia severity metric. And this measure can be used either as a predictor of future outcomes or as an outcome itself, for example, in a clinical trial. So what D accomplishes is it ensures that the intervention is having a functionally salient clinical impact. If you can change the D score, then the cognitive changes you've affected are in fact driving changes in functional status. This cartoon summarizes really about a decade of work, but it represents an actual model in ADNI, which explains 65% of the variance in dementia severity as measured by D. And these are all independent, statistically significant predictors of the D score. And even though we kind of conflate dementia with Alzheimer's disease, I'd point out that amyloid by PET imaging is only one of the multiple significant drivers of dementia severity in ADNI. And it has a moderately strong association and explains a minority of D's variants. So to fully understand the presentation of a patient, you have to maybe accommodate the impact of multiple independent dementing processes that in their aggregate sum to explain the condition of the patient. So I thought it'd be interesting to build one of these models for you. This is a model I've published, and it's a model of ethnicities effects on dementia severity. I'm sorry, this model isn't published. This homolog is published. But anyway, so we're going to be looking in TARC at the impact of Mexican-American ethnicity on biomarkers associations with dementia severity. And we'll be using a homolog I call DEQ, and that's what's in this publication. DEQ was engineered to be factor equivalent across ethnicity, so that when we work with this homolog in TARC, we can be confident that dementia severity as measured in a non-Hispanic white is identical to dementia severity in a Mexican-American at the very same D-score, even if the Mexican Americans have much less educational attainment or are being interviewed in Spanish and tested with measures in translation. So the DEQ homologue is indicated by 5 cognitive measures and 1 measure of caregiver rated IDL. It achieves an area under the receiver operating curve of 0.95 to diagnose AD in TARC, and correlates 0.99 with dementia severity as rated by clinicians on the CDR. So the first step here is to add a biomarker. So this is CD40 being measured in TARC at baseline in serum, and I've regressed it onto dementia severity. We're going to use dementia severity 1 year later at wave 2. So this is a prospective longitudinal model. And of course, CD40 can't explain all of these variants, but the model has to account for 100% of the variance in the variables at play. And so that creates a latent residual, which is the variance in dementia severity not explained by CD40. And this path is the variance explained by CD40. TARC has some issues with its biomarkers. There are significant batch effects, and so I have to adjust in TARC for these batch effects with some dummy variables. And then we're interested in the effects of ethnicity, not intervening comorbidities that might be associated with ethnicity. So I'm going to adjust the model for multiple other covariates here, including hemoglobin A1C, gender, depression severity, body mass index, ApoE, 4 burden, age, and gender. Then we're going to add some statistical measures of model fit. Basically, I could fit the data to any possible model. Multiple models are available. I just have to build it. So we can test the relative fit of competing models to try to find the models with the best fit to the data. And the 2 critical statistics, I would say, are the CFI here, which should be as close to 1 as possible, and the RMSEA, which should be as close to 0. Then I'm going to stratify the entire model on ethnicity, and then finally constrain the biomarkers association to be equal across both ethnicities. So that's the null hypothesis. If this biomarker has if ethnicity has no effect on this biomarker's association, then this constraint will hold true and the model will fit well. But if ethnicity does have an effect, the model will fit less well than an unconstrained model, and we can test that through change in the chi-square statistic. So it just so happens that this biomarker is impacted by ethnicity, and so we reject the constraint. And in non-Hispanic whites, you can see that the model fits well. The CFI is 0.93 and the RMSEA is 0.03. But there is no significant association between this biomarker and dementia severity in non-Hispanic whites, but it is significantly associated with dementia severity in Mexican Americans. So I've gone through TARC's entire panel of 120 biomarkers with this type of approach and have identified these 42 serum proteins as being related to dementia severity only in non-Hispanic whites and not in Mexican Americans. So that's interesting because these may represent dementing processes that are disproportionately impinging on non-Hispanic whites and that Mexican Americans are somehow protected from. But in contrast, there are only 4 that are significantly associated with dementia severity in Mexican Americans only. Now, there is some risk. This is an ad hoc panel. We didn't choose these biomarkers rationally. The models have chosen them for us. But I don't think that these are by accident. These 4 proteins are quoted together in the abstracts of almost 500 PubMed papers, and 3 of the 4 are listed together in abstracts in 112 papers, mostly addressing endothelial vascular impairments in humans and in animals. And so the model selection of these proteins suggests that there may be a common mechanism underlying their action in Mexican Americans. And that gives me some hope that I can define that process through a new latent variable, which we'll call the biomarkers of Mexican Americans. This is the variance in this panel of biomarkers which they share with each other. And you can see that none of them load strongly on this construct. So most of the observed variance in each one of these biomarkers really has nothing to do with what they share, but what they share, a common process indicated by all 5 here, is strongly related to dementia severity and only in Mexican Americans, not in non-Hispanic whites. So, so basically now you could use this latent variable as an object of study to try to figure out what is going on with this, what biological process does it represent, and why does it impact Mexican Americans and not non-Hispanic whites. This is intended to show that these biomarkers selected through D, or a latent variable in a structural equation framework, have real world consequences. So here is a multivariate regression model of my biomarker construct, BMA, and a bunch of covariates to try to predict dementia severity on the CDR as rated by clinicians. And this model confirms that the latent biomarker construct predicts CDR only in Mexican Americans, not in non-Hispanic whites. This is a second demonstration of the real world consequences of biomarkers that we develop through these structural equation approaches. So this is a multivariate logistic model of 5-year conversion from non-demented states to clinical dementia in TARC. So it's a prospective model over 5 years. And the usual demographic subsets are significant predictors of dementia, dementia conversion, incident conversion. The odds ratios associated with these independent predictors sum to a 9-fold combined risk. But for each one of them, I've developed mediation models of their biomarkers that mediate their associations with D scores. And so I have the short list of biomarkers from TARC's panel that mediate each of these effects. These are independent effects, and the biomarkers selected are largely non-overlapping. So what we can try to do is select a biomarker from this list and try to displace these demographic predictors as predictors of dementia conversion over the next 5 years. And you can see that I've accomplished that. I've fully attenuated age, gender, depression, and ApoE through the introduction of 5 biomarkers selected by SEM. ApoE's effect is uniquely and fully attenuated by C-reactive protein and inflammatory protein. Depression's effect is fully attenuated uniquely by resistin. It took 2 proteins to attenuate age's 3-fold increased MCI conversion risk, and gender's effect is fully attenuated by thrombopoietin. So these are 22 serum proteins that mediate gender's effect on D and TARC. And I'll just draw your attention to 2 of them, thrombopoietin and prostatic acid phosphatase. Prostatic acid phosphatase fully mediates gender's effect, and thrombopoietin mediates 53% by McKinnon's mediation analysis. So the reason I've chosen these 2 is simply because I know of FDA-approved medications that might be used off-label to modulate those biomarkers and thereby potentially attenuate gender's risk on dementia severity in clinical trials. This shows that basically these 2 proteins significantly predict dementia severity in women in TARC, measured in serum, but they replicate, those associations replicate to plasma in a completely different cohort, ADNI, where they are associated with dementia severity in women only. And this effect on dementia severity is independent, again, of amyloid PET SUVR. So I think it represents a gender-specific dementing process, not really something that dovetails with Alzheimer's disease. to do a clinical trial, we'd really want to know which women are adversely impacted by the targeted biomarker, in this case, PAP. And so I've developed an algorithm to address that. I'm going to take another homolog, which is called detail. Detail has the advantage of being engineered to be delivered over the telephone. So a lay interviewer can develop this detail score with about a half-hour interview over the phone. We'll take the detail construct and adjust it for serum prostatic acid phosphatase. Now, I showed you before that this biomarker has a weak effect on D, so the adjusted and unadjusted constructs correlate strongly. But if PAP had no effect, then there would be no difference in these two constructs, and every case would align along this line of identity. And you can see visually that they don't. Some cases to the left of this line are adversely impacted by serum PAP. The cases to the right of the line are protected by their serum PAP levels. And it turns out that almost 50% of women are adversely impacted by PAP. And these cases present along detail's entire range, which means women are being adversely impacted by PAP at every stage of dementia severity, from the very best normal performance all the way through to the very worst dementia case. And in fact, the groups selected by this algorithm differ significantly in their observed serum PAP scores, even though they were identified entirely by cognitive assessment, which potentially could be delivered by telephone. So the adversely impacted fraction have demonstrably higher serum PAP levels, and this can be done, over the telephone. And this approach can be done really with any biomarker. So by a single telephone assessment, I could nest these models over each other and screen candidates for multiple clinical trials by a single telephone administered cognitive assessment. And I'll just briefly run through some other biomarkers. One of the ones I showed you was a latent variable inflammation indicated by 10 serum proteins. This shows you that it's more strongly related to D, so more cases are off the line of identity. This is in serum, inflammation measured in serum in TARC. This is inflammation measured in plasma in ADNI. And here you can see that the groups selected as being afflicted by this cognitive assessment differ significantly in both inflammation measured in serum in TARC and in plasma in ADNI. And not only the latent construct, but on each of the individual indicators, each of the individual indicators that define that construct. So every single one of them is significantly different in the group that I've identified through a cognitive assessment. So this approach can identify individuals who have entire profiles of multiple biomarkers, and you can then triage them for clinical intervention. And it even works for genetics. Here's the same approach applied to the ApoE4 allelic burden. You can see that it clearly distinguishes groups by a dose effect. These individuals have no E4, these have one E4, these have two. It's clearly a consistent dose effect at all stages of dementia severity. It does not become wider as cases become more demented. So ApoE's effect looks to me like it's incurred well before dementia's onset and in a dose-dependent manner. And here you can see how well it identifies carriers of the E4 allele. It almost perfectly predicts that a subject is carrying an E4 allele from a cognitive assessment alone. No genetic testing is necessary. So what I see these analyses is doing is kind of reshaping, at least my opinion, of how Alzheimer's works. I'm painting plaques and tangles into a corner. The observed dementia severity is being modulated by multiple independent processes, each of which has a unique biological pathway. And I've shown you that we can attenuate gender, depression, age, and ApoE with different serum and plasma protein biomarkers. And these are also serum proteins. That limits the traditional Alzheimer's related biomarkers to just a fraction of dementia severity, variance, and opens up all of these other predictors for exploration through easy to acquire blood-based proteins, which have potential treatment options. So in summary, we can pre-select cases for treatment relevant biomarker profiles through cognitive screening alone, perhaps in the future delivered by telephone or remotely. The selected cases are empirically adversely impacted by the targeted biomarkers, and then these individuals can be triaged into clinical trials that purport to modulate those selected biomarkers, kind of a precision medicine approach. And then we can use D as an outcome measure to ensure that the cognitive changes affected are clinically salient. That's the end of my remarks.
Okay, thank you, Dr. Royall. Let me check first if I have any questions from the audience in Q&A. Doesn't seem so. Okay, then let me check the participants. Oh, yes, we do have some raised hands from Igor, of course. So Igor, please speak up.
Thank you. Thank you for interesting presentation. Actually, you described quite complex picture. And what would be a main effect that generate disparities between Hispanic and non-Hispanic? Is this biology? Is this risk factors? Or maybe this is something happened during a diagnosis for these subgroups? What would your opinion about that?
Well, it's probably not simple. You know, I showed you I only found 4 proteins in TARC's panel that mediate the association of ethnicity. That doesn't mean there might be other ones that our panel doesn't cover. It's not a proteomic panel, for example. You might find other ones. And there are demographic differences in the Mexican Americans with regard to diabetes, vascular disease, that sort of thing. I guess what I'm saying is that there are many paths to any particular objective level of dementia severity. There are many paths to that. And Mexican Americans may become demented through some subset. Non-Hispanic whites may be disproportionately vulnerable to a different subset. And so you'll never be able to impact dementia severity meaningfully with a monolithic single intervention applied to both groups. You'll have to, at some point, come to terms with this and develop a very personalized, individualized treatment prescription, ideally based on individual biomarkers of interest.
Okay, thank you.
Okay, thank you. I see Anatoly raised hand in his camera, but not in Zoom. Anatoly, please go ahead.
Okay, thank you very much, Donald. Very interesting talk. And I especially think that this delta variable is extremely important for the analysis. And one time ago, I think Konstantin was also involved in the analysis. We look at the so-called demographic correlated frailty models. They use very similar approach just to estimate this correlated frailty characteristics, and we found that heritability in this latent variable is twice as high than longevity heritability. Did you ever think or try to estimate heritability in this delta variable? Because if they are more heritable, then there is a good idea to study genetics of this variable, because you have more chances to detect genes which influence longevity, in our case, through frailty, and the other traits which you use in analysis through this delta variable.
Yeah, so, one of the advantages of working with dementia as a latent construct is it removes a lot of measurement error. And so you have more power to make genetic associations. So TARC is not a large sample, but it does have a GWAS panel. And recently, a group in Fort Worth in North Texas University have associated D with genetic variability in the GWAS and identified several candidate genes that are specifically related to D in a sample as small as TARC. And I think the reason they were able to find significant effects in such a small sample is because using a latent variable instead of a categorical outcome variable greatly improves your power to detect genetic effects.
Right, exactly. Yeah, thank you very much.
Yeah.
Thank you, Professor Royall. I agree. It's really important to mention like this latent constructs instead of like separate biomarkers, for example. It's really, really important observation and really important concept as well. So it's really encouraging. Thank you for your presentation, very interesting presentation.